Thanks everybody for coming to the Travelogue lunch. It is my great pleasure today to introduce you to the speaker. She's very close to our institution. Actually, Morgan is a research botanist here at the Botanical Research Institute of Texas and Forward Botanic Garden, and he's also the director of the Global Genome Initiative for Gardens. Morgan has been here since 2018. He has a bachelor in biology and a master's in biology from Virginia Commonwealth and a PhD from George Mason University. And today, Morgan is going to be talking a little bit about one or just the some of a travelogue of a trip that he did, but that is funded through a National Science Foundation grant of Ironweeds of the Eastern Hemisphere. So with that, thank you, Morgan. I'm very excited to hear about your adventures, and welcome everybody, and thanks for coming.
Thank you, Ale, for the warm introduction. It's exciting to be here today. Thanks everyone for coming. As Ale mentioned, I'm going to be talking today about my research here as a botanist at BRIT, particularly in the context of my current NSF-funded research project, focused on developing a subtribal classification and generic delimitation of ironweeds in the Eastern Hemisphere. So this project is focused on helping us advance our systematic knowledge of one of the most diverse groups, or what we call tribes, of sunflowers and daisies in the world, from this particular part of the world where they are especially diverse in the Eastern Hemisphere, from Africa to Southeast Asia. In November, as Ali mentioned, I traveled to South Africa, where we traveled over 4,000 miles to more than 20 different protected areas in search of as many of these species as possible, so that we could really lay a strong foundation for the start of our project. Now, before I get into the fun, enticing travelogue part of this, I'm going to try to teach y'all some things. So hopefully I've effectively enticed you to coming in hopes of seeing some pretty pictures, and you will see those, but first you're going to learn something, I hope. Before I begin, I just want to say some words of thanks to two very important people in my life and in my career. My late postdoctoral advisor at the Smithsonian, Dr. Vicki Funk, and Harold Robinson, Dr. Harold Robinson, both of whom were curators at the Smithsonian for more than 30 years. Sadly, we lost them in 2019 and 2020. They really laid the foundation for most of the work that I'm going to be presenting in my talk today. So I just want to express my gratitude for them in all that they did to introduce me to this group and train me to understand just how amazing these plants are. I also want to thank the organizations that have funded this project, the National Science Foundation of the United States, and the Natural Environment Research Council from the United Kingdom, who supported this project entitled Subtribal Classification and Generic Delimitation among Eastern Hemisphere Ironweeds. This is a 4-year project. It's funded at about $1.2 million. Right here at BRIT, we have 2 staff or students working on this project, myself as PI, principal investigator, and PhD student, Ando Andrea Manohera, who started at University of Texas at Arlington just last August. I also want to thank my esteemed colleagues at Royal Botanic Gardens Kew, Drs. Isabel Laridon, Benoit Loyal, who you'll see in a number of photos because he accompanied me on this trip to South Africa, Anahita Simoes, and Dr. Igor Antonio Dominguez. Before I begin, just an outline of what I'm going to go over. I've divided the talk into 3 main sections. I'm first going to provide some background and introduction. This is the part where I hope you learn some things. Then I'm going to go into details about the actual travelogue, highlighting this first expedition of 5, eventually, that will round out the entirety of this project. And I'm not going to just dive right into pretty pictures. I want to also talk to you about what it takes to plan a trip like this, and also what we accomplished at the end of the trip. And then lastly, I'll just quickly round out the talk by highlighting next steps and future objectives. So without further ado, whenever I give a talk, I like to explain to people what I do in my lab. I've been a botanist for over 15 years now, and anytime I introduce my work to other people, I like to tell them that I got my start, probably like anyone else, with my ABCs, but my ABCs might be a little bit different than others. I started with my master's thesis focusing on the ginseng or English ivy family, Araliaceae. I then moved on to my PhD. My doctoral dissertation research was in the frankincense and myrrh family, or Burseraceae, so the B of the ABCs. But most of my work really centers around the most species-rich family of plants on Earth, the incredible, the unimitable Compositae, the C of the ABCs. So this is the sunflower and daisy family that you are probably most familiar with, but I'm going to introduce you to some of the lesser known, but equally famous and beautiful members of this family today. Compositae is an incredible model for myself and other botanists who study it for a number of reasons. First of all, I already sort of spoiled part of the punchline. It is the most species-rich family of plants on Earth. There are over 30,000 species of Compositae, which means that almost 1 out of every 10 species of flowering plant or angiosperm belongs to the family. They exhibit incredibly diverse morphologies, as shown by these photos here. They have a diverse assortment of life histories, and you can find them nearly everywhere on Earth except for Antarctica. So no matter where you are or who you are, you can jump in and start working with this family. And I'll tell you, with that many species, we welcome the help. Now, you might not realize it, but you probably recognize many of the members of this family. They include a number of economically important crops, such as chamomile, Berbera daisies, stevia, artichokes, sunflower seeds, of course, lettuce, and safflower oil, among many others. And it's also an exciting time to be a researcher who focuses on Compositae, because there are a number of rapidly expanding resources that are increasingly becoming available for us, those of us who study the family. This is partly driven by a diverse and international and growing international research community through the International Compositae Alliance, or TICA. It's also facilitated by an increasing number of molecular markers that are facilitating comparative phylogenomic research that is allowing us to reconstruct the evolutionary history or family tree of all of the species that belong to this family. Lastly, there's also been significant growth in the number of sequenced whole genomes for this family. This has been stalled because the genomes in this family are extremely large and complex. And so it's taken some time for us to be able to actually sequence these and assemble them in a way that's useful for researchers around the world. This is depicted by the graph here, which shows the exponential growth in the number of sequences that are available in GenBig just over the last 10 or 15 years. Whenever I give a talk on comps, I like to sort of ground the audience because 30,000 species is a lot to think about. And the best way to ground the audience is to help them understand that even as somebody who specializes in this family, I have to ground myself. And to do that, I think about the classification of all of these species into smaller groups. And in Compositae, we organize all of these species into smaller groups that we call tribes. There are about 50 tribes of Compositae. And so I've worked in a couple of these tribes. I'll talk about these in a moment, and I'll end, of course, with the group that I'm going to be talking about today, the Vernonieae. But this is a publication, this is a figure from a publication by Alfonso Susana and colleagues in 2020 that is a classification of the entire family. This was a posthumous tribute to Dr. Vicky Funk, my postdoctoral advisor, and this is really her ultimate, her final classification for the family. All of the circles that you see depicted in this figure correspond to one of these 50 tribes. The size of that circle corresponds to the number of species in that tribe. So some of these circles are very large, while others correspond to only one single species. There are some species that are so unique, different, distinctive, that they're the only species known from that entire tribe. Some of the groups that I've worked in include Gostel, Tarconanthe, Dicomi, Neuralaene, Tajidi, and of course, what I'll be focusing on today, the Ironweeds or Vernonieae. You'll note that the circle that corresponds to Vernonieae is rather large, and Vernonieae is in fact the 6th largest tribe by species number in the entire family. Luckily, they're pretty easy to recognize. Vernonieae can be recognized by a number of shared characteristics, including the presence of alternate leaves. what we call homogamous capitula. All of the florets on the capitulum, or the head of the flowering structure in these plants, are bisexual. We also have a multiseriate involucre. The involucre I like to describe to a general audience as sort of like the protective sheath that surrounds the developing flowers before the capitulum opens. That protective sheath, or the involucre of bracts that protect those flowers, will open up and reveal itself. Multiseriate just means that the number of bracts occur in a series. So I won't get into too much detail because I know we have a partially general audience here today, but this is really helpful for botanists. They also have mostly violet, pale violet, or white flowers, corollas, with especially narrow style branches. Styles are the female reproductive part of the flower, and they have acute apices, or their tips are pointed. So this, believe it or not, makes it pretty easy to identify this tribe using this handful of characters. And this is what we commonly employ as botanists who specialize in this family. We have to look at a number of morphological traits, features of these plants that combine together to make sort of a recognizable unit of diversity that we call tribes. And so this is that suite of characters that we use for Vernonieae. Now, unfortunately, for much of Vernonieae's history, it has received a number of pejorative adjectives by botanists who have described it. One of these is the evil tribe. That doesn't sound so appealing, does it? So you're probably wondering if I'm some kind of masochist for getting involved in research of Vernonieae. But one of the reasons it's been referred to historically as the evil tribe is because there are simply so many species. There are about 1,500 species in this tribe, and they're grouped into about 130 genera worldwide. As you can see, diversity of Vernonieae is almost evenly divided between the Americas, or the Western Hemisphere, with 13 subtribes, 69 genera, and about 800 species, and the Eastern Hemisphere with 6 subtribes, 67 genera, and about 700 species. A number of the reasons for calling or referring to this tribe as the evil tribe has to do with what botanists have historically referred to as the Vernonieae problem. And the Vernonieae problem simply tells us that for much of the history of this tribe, a majority of species in the tribe, over 1,000, have been recognized in a single large genus called Vernonieae. Now, all of the species that are recognized in this genus are recognized historically largely on the basis of characters that they do not share, rather than on the basis of shared characteristics that unite them into an easily recognizable genus. So for this reason, Vernonieae, unfortunately, for over 200 years, has been recognized sort of as a waste bin genus. If a botanist collected a plant, knew that it was Vernonieae, but didn't know what genus it belonged to, go ahead and put it in Vernonieae. So that Vernonieae problem has persisted for centuries until Harold Robinson, this botanist and curator at the Smithsonian I mentioned at the beginning of my talk, reduced the size of Vernonieae to recognize only about 20 species, largely from North America. So, taxonomically, what happens when Harold did that? Well, when Harold reduced the size of Vernonieae to only about 20 species, it immediately put into play a series of activities that botanists and taxonomists needed to then undertake to help figure out where all the 980 or so remaining species that previously were identified in Vernonieae, where they actually needed to be placed. Now, much of this work has been completed by botanists for the species that were formally recognized as Vernonieae in the Americas. And so now we have 69 genera that account for a majority of those 800 species. The situation is not so tidy and not so neat for the species that exist in the Eastern Hemisphere, because unfortunately, botanists have not paid as much attention to the species diversity in this tribe from the Eastern Hemisphere. This is really an important part of why we received the funding for this project, because our goal is to establish the foundation that will allow us to place all of these now orphaned species that formerly belonged in Vernonieae into their correct segregate genus. So Vernonieae in the Eastern Hemisphere, as I mentioned, is very diverse. There are 6 subtribes, 67 genera. Shockingly, over 30% of these 67 genera are what we call monotypic. That means these genera only contain a single species. And this has been common practice for a lot of these groups in Vernonieae historically, because botanists and specialists who work with this group would identify a species that is especially distinctive, that didn't really look like any of the other species in any of the other genera. And for this reason, they've described a new genus for that species. That is fine, but it's led to some very complicated work for us as taxonomists to figure out where all these other species actually belong. And there remain about 200 species in Vernonieae from the Eastern Hemisphere that still carry the name Vernonia and need to be placed into the correct segregate genus. As you can see, species from the Eastern Hemisphere, these are all photographs that I've taken from Madagascar and South Africa, are beautiful. They're extremely diverse. They have all kinds of different life histories, habits, everything from teeny tiny little rosettes, herbaceous annual plants, to 20-meter tall trees. It's incredible the diversity that you see in this genus here, in this tribe here, all over the Eastern Hemisphere. And it's shocking to me that it has received so little attention by botanists. So when we set out to actually undertake these activities associated with this grant project, we outlined 5 key objectives to achieve our goals. And so I'm going to briefly go over those now. First, we needed to expand collections, especially from key priority areas that represented collection gaps in our data sets, both from historical collections and to facilitate our new research using novel molecular methods. So we plan to carry out field work that would generate 6,500 new collections from around the world in the Eastern Hemisphere, and also visit historical collections at natural history museums in herbaria to study about 50,000 sheets that correspond to species diversity of ironweeds. Our second key objective was to develop a robust evolutionary framework in order to guide our taxonomic revision that needs to take place. And this will be carried out using an integrative approach that combines molecular sequence data with comparative morphological data. I'll talk about that a little bit later. Third, we aim to place all of those orphan taxa, all of those orphaned species of Vernonia that I mentioned, about 200 of them, into their correct segregate genus. We then plan to disseminate all of the results that we have gathered through what we call the Global Composite Database. This is an online and publicly accessible database and taxonomic resource available to all. And lastly, train the next generation of composite taxonomists so that we can ensure that what we're doing as botanists and as taxonomists builds on a legacy that I have fortunately benefited from, some of the best, Vicky, Harold, and others, and carry that torch forward so that in the years, decades, and centuries to come, botanists will have a strong foundation and knowledge base to lead this work going forward. So just to give a little bit more detail on each of these priorities, I'm going to first focus on our plans for field work in our area. Today, I'm going to be talking about the first of our field work trips. As I mentioned, we're aiming to collect about 6,500 new collections from the field. Our first trip, as I mentioned, was just in November to South Africa. We have 4 other trips planned, one to Central Africa, one to Mozambique, one to Madagascar, and one to Thailand. And we also have trips planned to 5 of the herbaria that we have identified as having the most important collections for us to study historical data and collections of this group. This includes the National Herbarium in Brussels, Belgium, Royal Botanical Garden Kew's Herbarium, the Missouri Botanical Garden, the Paris Herbarium, and the US National Herbarium at the Smithsonian Institution. Our second objective here is, as I mentioned, to develop a robust evolutionary framework in order to guide our taxonomic revision. And I mentioned that we're going to be combining molecular with morphological data. So what does this look like? Well, we've identified 2 important molecular sequence data sets that will help us with this particular task. that correspond to two sets of molecular loci that can be used to reconstruct and test evolutionary hypotheses. This is the Angiosperms 353 dataset and the Composite COS or Composite 1061 dataset. We will use those data to reconstruct these relationships in combination with a suite of morphological data that we will measure when we carefully review historical specimens from herbaria all over the world. So we will be generating a database of morphological characters, both macro and micro morphological characters, in order to test hypotheses about whether this genus is distinct from this genus, how this genus moved across the planet, so on and so forth. What are micro characters? Well, micro characters, a great example is pollen. Pollen is teensy, eensy, weensy, right? It's tiny. You need a scanning electron microscope really to see it in detail. So that's a great example of a micro character. And one of the outputs of this project is going to be an atlas of the pollen of Vernonieae. Pollen is well known as being highly variable in Vernonieae. And in fact, this tribe has some of the most diverse pollen types of any group of plants on Earth. So this is a great system to test the efficacy of pollen morphology for taxonomic purposes. To zoom in a little bit on what I mean by developing a robust evolutionary framework for guiding our taxonomic revision, I want to give an example of a paper that was just recently last month accepted for publication in the Botanical Journal of the Linnaean Society. This corresponds to the most comprehensive sampling of Vernonieae from the Eastern Hemisphere to date, and includes 102 taxa and 70 loci. As you see here, this is just, well, what is this? It looks like a comb. Did I brush my hair with this today? I don't know. Well, this is a depiction of evolutionary relationships as inferred using molecular sequence data. And when we actually plot onto this comb, onto this evolutionary tree, the names of these groups, what we see is strong support for a monophyletic, a clade that corresponds to core Vernonieae. So these are the usual suspects, what we expect when we see Vernonieae. But then, surprisingly, we saw that one of the genera, one of the largest genera in Vernonieae, actually with 43 species, Distephanus, is not falling out in the same group. It's not falling out with the rest of Vernonieae. And we realized that it's actually sister to another group that includes Mochini, which is a very small tribe, only from South America, and core Vernonieae from all over the world. So this tells us Distephanus is a distinct evolutionary lineage, and in this paper, we're describing it as a new tribe. So we have one more new tribe to add to the taxonomy of Vernonieae, of Composite. So our third objective I mentioned is we seek to place all of the orphan species of Vernonieae into their correct segregate genus. And unfortunately, a lot of the time right now, we simply don't know what those segregate genera are. We don't know where they're going to end up because we haven't done the work. We don't have the data to actually tell us where they belong. So I'm going to use the same example to illustrate what I mean by this. Two species of Distephanus that were sampled in our phylogeny here, we found do not actually come out in Distephanus. We actually find that they're placed in the core Vernonieae over here in the purple, where those two red arrows are. This wasn't particularly surprising to me. They were included in Distephanus because they share trinervately veined leaves. However, as you can see, they have violet flowers. All other species of Distephanus have yellow flowers, or cream-colored flowers, or orange-colored flowers, but none of them have purple flowers. So as it turns out, these are indeed part of Vernonieae, but we don't know where to actually place them. We don't know what genus these are going to end up in right now, because we simply haven't sampled enough. And we don't have morphological characters that can confidently assign them to an alternative segregate genus. So for now, these two species, Distephanus anguillifolius and Biafri, will remain in Distephanus as sort of orphaned species that will await future work so that we can confidently assign them to their correct new genus. Our fourth objective, I mentioned, is to disseminate our taxonomic information and our results through the global composite database. This is an online, accessible, publicly accessible portal. It's a permanent resource that we hope will serve as a gold standard for taxonomic databases. It was established in 2019, and it's recognized by the World Flora Online as a taxonomic expert network. It was built using the foundation of the infrastructure of the World Register of Marine Species, worms. And as such, it does come with some complications. Obviously, compositae is not a group of marine organisms. They're terrestrial. And so a lot of the terminology in this database is still rooted in terminology that we associate with marine organisms. Are they benthic? Are they blah, 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 whatever. And so this is some of the updating that we need to do to this database to make it more relevant and informative for taxonomists who study and work with this group. This is essentially like Wikipedia for plant taxonomists, and the only difference is that it is an authoritative taxonomic resource because only specialists who are taxonomists and who are verified can contribute data to the global compositae database, as opposed to Wikipedia, where anyone can get online and start editing Wikipedia pages, right? So this is an expert curated database that provides taxonomic reference information for the entire world. Lastly, I mentioned our fifth goal is to train the next generation of compositae taxonomists, and this includes both students and professionals. So we have 2 master's students and 1 PhD student who will be supported through this project. PhD student, Andrea Monahera, is sitting with us here. She just started at UT Arlington in August. Very lucky to have her. She is an incredible student. We also will be training a lab technician as well as hiring a postdoc. Last March, we hired our lab technician, who is our resident pollen specialist. He's a palynologist. In 1 year, he's just done incredible work. He's already published our first paper on this project, which came out last week in the journal Grana, highlighting a synopsis of the pollen of a really distinctive genus in this tribe, Vernonieae. We also train each summer 1 summer undergraduate student. And so last summer we welcomed Iris Schmieder, who had just graduated from high school, did an incredible job, and is now an undergraduate student at UT Austin. We also are training the next generation by offering workshops that are associated with each of our field expeditions. So we're training the local collaborators that we work with on the ground when we're doing field work by hosting workshops and boot camps. We're also conducting public outreach and community science, and we're presenting our results at national and international conferences. That rounds out the first part of my talk. So now we can get into the good stuff that y'all are probably hear, pretty pictures. Before I get into that though, I just have a little bit more background for you. I want to talk about what it takes to plan an expedition like this, because it does take a lot of work. Whoever you are, it doesn't matter. You really need to do your homework, and that usually involves consulting historical taxonomic literature, as well as historical collections at natural history museums and herbaria. So for us, that historical literature looked like this, going back to the mid-19th century, looking at Flora Capensis by Harvey and Saunder, looking at some of the more recent publications, for example, by Harold Robinson, who published in 1999 a partial revision of Paleotropical Vernonieae, more recent work by Nononuleko Swalankamo, a South African botanist, on 2 of the genera of Vernonieae from South Africa, and a capstone paper by Harold Robinson and colleagues in 2016, highlighting the diversity of Vernonieae from Southern Africa. All of this to us pointed to South Africa being the best launch-off point for this project to start our fieldwork, because to date, it has the most comprehensive data available to us and most up-to-date data. South Africa also has an incredibly rich history of having wonderful botanists who have contributed to our understanding of the flora of South Africa. And so this really made it a great starting point for our project. So on the other side of the planning equation, we have data, right? So there's historical literature, and then thanks to advances in computational sciences in the 21st century, we're able to look at data from natural history collections all over the world that has been digitized through projects like the Torch Herbarium project here at BRIT. South Africa has its very own botanical database of Southern Africa, or BODATSA, and you can access this data online anywhere in the world. through the Global Biodiversity Information Facility, or GBIF. So this is a plot of all of the data that is in BODATSA through GBIF. It's a heat map that shows the location of records that are in the collections of herbaria in South Africa. You can go into that data, filter it to only show you records that correspond to Vernonieae, for example. And then when you do that, you can download the data and you can plot it in interesting ways that can help you plan a field trip. So that's what we did. First thing we needed to do was really figure out, okay, when are we going to go to South Africa? And when we plotted this data, we found that the months of October, November, and December were the ideal times to maximize the potential collection of species in this tribe, right? You do see that they flower, they're available all year based on the collections data, but November certainly stands out as an outlier here as being especially rich for us. So we use this data to plan our trip and the timing of our trip, specifically in November. But that's not all we did. We also downloaded this data, and luckily much of it is geo-referenced. So there are GPS coordinates associated with many of the collections in BODATSA. And if we overlay those onto a map of South Africa, we can identify areas of priority collecting. And when we did that, we realized that clearly northern and eastern South Africa was the prime spot. That's where we need to be focusing our attention. South Africa is a big country, and we need to efficiently utilize our time and our cost resources. I didn't just plot these on a map though, because these records correspond sometimes to collections that were made over 100 years ago. And sometimes when you go to a place where a plant was collected 100 years ago, there might be a shopping mall, there might be a new neighborhood development, there might be a parking lot. So another added layer of planning and contingency to make sure that what we're looking for, we're going to hopefully find, is to overlay on this map a map of protected areas. These are national parks, these are natural reserves, and other similar areas where we can at least hope that the native flora has been preserved and protected. So it hasn't been developed and bulldozed over. And so using this combination of geographic data, we were able to not only pinpoint the time that we would go to South Africa, but exactly where we would want to go. At the end of the day, what this looked like was an expectation that we would probably collect, if we traveled in November, 9 genera and 27 species. At the end of 25 days, driving over 4,000 miles across South Africa, we actually exceeded our expectations. We collected 10 of the 15 genera of Vernonieae that is known from South Africa and 29 species, so this was pretty cool. The planning equation is not just data. I would actually argue the most important part of any planning for a trip like this internationally is to work with local collaborators. And luckily, we had some incredible local collaborators to work with, most of whom were based at the South African National Biodiversity Institute, SANBI, and the National Herbarium in Pretoria. These include our collaborators, Dr. Miranda Kokemer, Nicola Berg, Eric van Wyk, Peter Bester, and 2 other colleagues who accompanied us in the field. One of whom I can't thank enough, Truus van der Merwe, who is a freelance botanist. He owned a nursery for 18 years. He walked away from the nursery business and decided he wanted to go out and just study and understand the diversity of plants in South Africa. He is one of the most knowledgeable people I've ever met anywhere with regard to understanding and knowing the local native flora of any place on Earth. And we could not have done this trip without him. So thank you, Truus, if you're watching. He's, as I mentioned, a freelancer. Our other collaborator who joined us in the field, Winston Woudenberg, who's based at the National Botanic Garden of Durban in the Natal province, accompanied us for about half the trip. And he was equally just an incredible resource to have along with us. So huge thanks to this entire group of folks for facilitating our trip. Now, at the end of all this planning, we realized that in South Africa, there are 42 species of Vernonieae that belong to 15 genera. And that data kind of looks like this. So the taxa that we were actually able to collect after 25 days are highlighted in blue. Again, we were able to collect 29 out of the 42 species, which is pretty darn impressive, and 10 out of 15 genera. it would be impossible to go at any single month in the year and collect all of these genera because they simply don't flower, they don't fruit, and sometimes they don't emerge at all at the same time. And so we're fighting against different flowering seasons. So for all that we could have done, this is absolutely the best possible outcome for this trip, and we were super impressed. So for the rest of the talk, I want to invite you to come along with me, board a plane at DFW, and let's hop on over to our first stop at the Pretoria National Herbarium. When we got off the plane in Johannesburg, we were greeted with, I gotta say, the best greeting I have ever had when I started a field trip. There were crowds outside, waiting at arrivals, they were singing, playing music. South African Broadcasting Channel was there. They interviewed me when I got off the plane. It was incredible. And it just so happened that an hour after our plane arrived, the national rugby team were jumping down with the World Cup that they had just won. So maybe it wasn't for us, but I think it actually was. I think we were getting a little good start and good luck for our trip, thanks to all these fans of botany in South Africa. So when we hit the ground in the herbarium, we started to really look in earnest. This was our first time, many of us meeting each other in person for the first time. We were going to be spending a lot of time in a car together. We wanted to make sure our plan was ironclad, super tight. So this is what our Google Maps route looks like. It's about 38 hours of driving, and this is not complete. And that's if we were just to go continuous in one loop. I maxed out the number of points that you can put in a Google map, so I just kind of like summarized it. At the end of the day, as I mentioned, it turned out to about 4,000 miles that we drove over 25 days. We brought along some helpful supplies, including a do-it-yourself DIY plant dryer that I made using supplies from the Home Depot, and an electric dryer. So that all the plants that we collected every day, if we were staying somewhere that night that had electricity, we could plug in that dryer and dry those plants and bring them with us. Midway through the trip, we could bundle those, put them in a like UPS basically, and ship them to the National Herbarium so that we could free up some space in our vehicle, which we dearly needed, despite the best efforts of our packer here, Miss Ostrich, who was doing all she could to make sure we had extra space in our vehicle, looking for all kinds of snacks in there that might have fallen out. But after our first team dinner, we went ahead and hit the road. So our first stop took us from Pretoria National Herbarium northward near the border with Zimbabwe, about 20 miles from the border with Zimbabwe, a town called Messina in the South Pansberg. That's an Afrikaans word that just means the salt pan mountains. Here we encountered a lot of wildlife, of course. Many people, when they think of South Africa, they think about the incredibly rich diversity of wildlife. But we also saw some incredible landscapes. And my personal favorite is the largest and potentially oldest baobab in the world, which is kind of like my spirit tree. And if you want to get an idea of how big this tree is, that's me right there. And that's the tree. So that's about a 3,000-year-old baobab. Pretty cool. That was one of my highlights from the first stop. The real highlights though, and this is the formula I'm going to show for the next 14 slides, is I'm going to show a little bit about the site where we collected, and then I'm going to show what we actually collected, what plants we were after. So I want to feature two genera here. First is Hilliardiella. This was described by Harold Robinson, this genus, in honor of an incredibly important figure in South African botany, Olive Mary Hilliard. She is among all women botanists in the world. She is ranked fifth among women who have described new species of plants in the world, ever. She is an incredibly amazing person. If you want to read an interesting history about a botanist, look up Olive Mary Hilliard. She is fantastic. We know that Hilliardiella is represented by 8 species across South Africa. And over the course of our trip, we were able to collect all 8 of them. So that was pretty awesome. That's the first time I've ever done that. Ever collected all species of a genus from an area if there are more than 5 species of that genus in that area. So this was a first for me. It's pretty cool. The second genus, Orbivestis, is only represented by a single species in South Africa, Orbibestis cinerascens, and in particular is only known from this northern area near Musina, where we were doing this first leg of the trip. So we were really lucky to find this in flower as well. I also want to, after our first stop, we moved a little bit southeast from Messina to Salpansberg to the Volkberg wilderness area. So if you'll join me as we move along a little southeastward. This is characterized by an extremely heterogeneous landscape. It's sort of the intersection of a number of different mountain ranges. So you have overlap a little bit with the Salpansberg, the Salt Pan Mountains, with the Volkberg. It leads to just some breathtaking landscapes. And for me, one of the coolest things is sometimes you're standing in a dry forest and you can look about 5 miles in front of you at a mountain that is a dense, humid tropical rainforest. And seeing the juxtaposition of those two landscapes and habitat types right in front of you, I mean, you can look at it on the map and you don't really understand what it translates to, but when you see it in person, it's just remarkable to see completely different types of plants growing 5 miles adjacent to one another. It's just awesome. So here's some of the success, a joyful Benoit running back to us with our target species here, Hilliardiella hirsuta from the Volkberg. There were 2 key genera that we were interested in collecting here, one of which was Cyanthillium, which is represented by 3 species in South Africa. We collected 2 of them, and Lindsea, which is represented also just by a single species. Lindsea is very easy to recognize. It almost looks like those filaries I mentioned in Bolliger are like a sawtooth. So they have these spicules on the margin of the filaries. So it's a very easy character to recognize this group by, but it does take a pretty well-trained eye to know what you're looking for, I guess. Moving along from site 2, we moved from the Volkberg to Blind River Canyon and the Barberton Ranges. I'm gonna sound like a broken record, but it's just landscape after landscape after landscape that takes your breath away. This is a photo from Blind River Canyon, and this is Vernonieae here at the 3 roundabouts, which is one of the top sites at Blind River Canyon. We're standing about 2 feet away from the collection of Oocephala, one of the genera that we collected at the site. Blind River Canyon is one of the largest canyons in the world, up there with the Grand Canyon, which is obviously larger, but just a couple below it. And the Barberton's claim to fame is that the Barbertons are home to some of the oldest exposed rocks on Earth, almost as old as the planet itself, at about 3.5 billion years old. So this was a pretty cool area to go botanizing in. Not a whole lot of wildlife here. We did see some pretty cool stuff like circles, which I didn't get a photo of, but anyway, we're not here for that too much. Oh yeah, zebras, ostriches, more buffalo. But the genera, let me tell you, this is pretty cool. So Gymnanthemum is the genus that our PhD student working on the project focuses on, specializes on. Andu here, there are 9 species that occur throughout South Africa. We were able to collect 8 of them. So that was really exciting for us. And we were also able to collect this really narrowly indented genus, Oocephala centarioides, which was growing right on the edge of Blind River Canyon. There are only 2 species in South Africa, and we fortunately got both of them. We move along from the Barbertons, moving southeast. We're going to the coast now, to Isimangaliso Wetland Park and St. Lucia. This was a really cool area for all kinds of reasons. We got exactly what we were looking for there, but we also had some interesting neighbors. So you can't hear it here, but this is one of our neighbors 2 doors down from where we were staying in St. Lucia. That's a hippopotamus that's as big as our 4x4, just munching on our neighbor's grass as a reminder not to go wandering around at night. If you want to do a really great game drive, I encourage you to go to Isimangaliso Wetland Park and Shlushluwe Nature Reserve, both of which are just incredible. We saw so much wildlife, which was wonderful. Of course, we're not there looking for wildlife, but it's also really freaking scary sometimes to get out of the car and go collect plants when you're looking at a warthog as big as you. Or, you know, going into the water to collect a little Ethulia like this one here, which is right on the bank of St. Lucia Lake. And it's completely pitch black water surrounded by crocodiles. We did our due diligence. We were careful not to, you know, look for any disturbance, but you really never know. So it is pretty scary sometimes. We were able to collect this one species of Ethulia. This was key. This was a big one. Ethulia is very easy to recognize. Most daisies and sunflowers have a pappus on the fruit, right? It's that fuzzy, hairy thing that, like a dandelion, when you blow it, it lets the seed disperse. Well, it's absent in Ethulia. So the fruiting structures look really weird, and they're very distinctive and easy to recognize. We also collected 2 species of Distephanus from this park, 2 of which that we did not have in our sampling, so those are really key. And again, they were very easy to recognize based on this trinervate vegetation that you can see here of the leaves and these yellow flowers. From Isimangaliso, we traveled south along the coast, past Durban, to Insuleni and Vernon Crooks nature reserves. These were some pretty cool surprises. These were smaller nature reserves. We saw a lot of really spectacular wildlife again, but my favorite is going into these grasslands and seeing like this Gerbera daisy, and it doesn't come through very well in this photograph, but pockmarked everywhere in this photograph are daisies, just popping out everywhere, yellows, golds, purples, white. It's just incredible, this like symphony of color in the grassland. And then on the horizon line, of course, you can see wildebeest, zebras, antelope, just watching you, wondering what you're doing. It's a strange feeling, I've got to say, but after time, you start to get more comfortable with it. We did collect 2 repeat genera here, but they were really important species. One was Distephanus anisacatoides. Distephanus in South Africa is this kind of like scraggly, scrappy shrub thing. And it's too small to kind of support its own weight as it continues to grow. So the way it actually starts growing is it flops over onto the vegetation that grows adjacent to it. And it just starts climbing along the top of adjacent vegetation. So you get these really cool mats of Distephanus that form these bright yellow gold trumpets like of flowers that are escaping out of the top of all the adjacent trees and shrubs that it's growing with. It's pretty cool. It was really confusing to us because we were trying to figure out, is this thing actually a vine? Or is it a shrub? Or is it a tree? Like, what is going on? How is this thing growing? And we realized it's just this shrub that grows up like a little stick and then falls over and keeps going. So it's kind of cool to see how plants can figure out how to survive sometimes. Gymnanthemum from Vernon Crooks, we got this really our last species of gymnanthemum that we were after, Gymnanthemum crataefolium. Very distinctive, something that we had spent half the trip wondering, like, have we already collected this? And we just don't know it, because we don't really know what it looks like. And we finally saw it and we were like, oh my gosh, thank goodness, we did get it. This is it. It's totally different from the other things. Great. So you have these moments of like excitement that, I don't know, it's hard to explain. From Vernon Crooks, we went westward to the Dragons Mountains, the Drakensberg. So Maloti Drakensberg Park is a UNESCO World Heritage Site. It is just like, I think there must be the highest density of postcards from South Africa, probably come from the Drakensberg. Again, it's just breathtaking landscape after landscape, incredible to collect here. You also have this amazing difference in the vegetation and the flora as you go up in altitude. So we ended up having to go to the border post on the border of South Africa in Lesotho and stamp our passport, drive up in 1.4 kilometers, you climb 1 kilometer in altitude. So you can imagine what that road looks like, and enter Lesotho because you're missing 1,000 meters of elevational change where specific plants are endemic to these little narrow bands of elevation. And if you don't hit those spots, you're not going to collect them. So that was pretty cool. Some of these crowned cranes here, just, yeah, beautiful thing after beautiful thing. This area was really important because there are two narrowly endemic species of Hilliardiella, and importantly, our last two on our list. So we were able to collect these Hilliardiella flanagani and nudicollis. They're these little scrappy, tiny little things with a basal rosette of leaves that you almost miss if you're not looking for it. But luckily we got it in flower. It was a very happy day, let me tell you. From Maloti Drakensberg, we continued north and skirted along the perimeter of the Drakensberg to Royal Natal National Park. Royal Natal National Park, again, like I said, it's just postcard after postcard kind of scenery. Maybe not this as a postcard image, but these buzzards eating a giraffe carcass here. We also saw a jackal during the day, which is pretty rare, going after this recently deceased giraffe. Another just beautiful place. This was really our last main stop on the trip. And we were after our one remaining missing species, Pseudopegalettia thodi. It's a little tiny annual herbaceous perennial plant that's about the size of my cell phone. And it's only been collected about 10 times. It's only in Bodazza, like 10 records. And it's in our naturalist about 4 times. So we had some little inclination of where we might be able to find the species, but we didn't know exactly where. We spent all day hiking in Royal Natal under full open sun exposure, exhausted. We didn't find it. We were pretty disappointed. We got to our lodging, no power, no food, nothing. We were kind of disappointed, let me say. And it was the last thing we were after. We woke up that morning. We asked the owner of the lodge where we were if we could explore his property a little bit because the habitat was right. And about 10 minutes later, Truus sort of shouts over to us, "Hey guys!" And sure enough, he's found it right at his feet. And so that was pretty exciting to find that last target species on our trip. There are only 2 species in the entire genus. We collected both of them. Same goes for Parapolydora. There are only 2 species and we got both of them. So pretty darn exciting trip. That concludes most of the travelogue I wanted to highlight. So this brings me sort of to wrapping up the expedition. In total, again, we collected 29 of the 42 species of Vernonieae from 10 of 15 genera. That brings us back from Royal Natal National Park to Pretoria National Herbarium, where we were able to take the collections that we had already dried in the field and compared them to collections that we were able to use to confirm our identifications. And then we were also able to give, as our last day at the herbarium, a workshop to provide a synopsis of the diversity of Vernonieae in South Africa, as well as some training workshops on how to use the Global Composite database and another program called Vernonieae. So that was very nice to at least try and get a little bit back to our wonderful local collaborators who had helped us so much during this month. That brings us to the last part of the talk, and that is next steps and future objectives. So as I mentioned, this project has funding for 5 total field expeditions. South Africa was just the first. We originally planned to do fieldwork in Democratic Republic of the Congo. That's a little up in the air due to the ongoing civil war in the eastern part of DRC. So we may have to pivot on that a bit, but we'll also be traveling to Madagascar, Mozambique, and Thailand, sort of replicating the formula of this trip in these other countries. We'll also be traveling to a number of those herbaria that I mentioned that are really important for studying certain plants. On Friday, actually, Ando and I are going to be traveling to St. Louis, to the Missouri Botanical Garden, where we'll spend a week during spring break. I wish a month. Working with the collection there. Missouri has an incredible collection of plants from Africa and Madagascar, and so that's really important for us. And lastly, we are just starting to get ready to do a lot of sequencing. So last week, Ando and I were in the lab doing DNA extractions. When we get back from Missouri, we're going to have a whole boatload of samples waiting for us that have just been dropped in the mail that we'll need to extract and send off for sequencing so that we can see what in the world is going on in the family tree of this particular tribe. When we take that molecular data, we're going to do some 23andMe and analyses with it. All that is to say, this is really just the tip of the ironweed iceberg. There are a lot of important reasons to do this work that I've outlined today. It lays the critical foundation for the base of all of our knowledge about this group of plants, but that's not all. This work also enables us to carry this work further with broader impacts. For example, understanding agricultural development of economically important species like Gymnanthemum amygdalenum, which is also known as African bitterleaf. You can probably go to a West African market or store here in the DFW region and find this in the freezer if you want to try cooking with it. You can probably look up some recipes if you'd like. It's used to lower blood pressure. It's used as an anti-malarial. It's sort of nature's pharmaceutical medicine cabinet. Centropalus pauciflorus is an important oilseed crop that is actively in the process of being domesticated. So this is an opportunity to understand more about the biology of these plants and the relationships of this genus to other genera in Vernonieae, to potentially explore other species that might have these beneficial properties. We also see an opportunity to understand the development of pollen diversity, as well as the evolution and maybe maintenance of reproductive barriers that might be associated with these diverse types of pollen. I mentioned Vernonieae has some of the most diverse types of pollen of any group of plants on Earth. And so understanding the role and function, why those different types of pollen have evolved is really curious to us, and whether or not that plays a role in reinforcing barriers to reproduction is a really important evolutionary question that we seek to explore. And lastly, Vernonieae is also well known for having extreme diversity of secondary chemicals that they produce as a defense against herbivores that want to eat them, insects that want to eat them. These are super potent chemicals sometimes. They're known to lead to the cause of death of cattle in South African relatives of this group. So they're extremely potent, but they're also potentially a treasure trove of medicines and pesticides and natural remedies that could be really impactful for improving the human condition, right? So I'm curious about understanding the evolutionary arms race that takes place between these species of Vernonieae and the herbivores that are trying to eat them. So as they're actively engaging in this chemical warfare. That being said, I have a lot of folks to thank, most importantly, Vicki and Carol for just introducing me to this group and helping me fall in love with it. But also our incredible collaborators. Truce, thank you so much. You are just amazing. We couldn't have done this trip without you, and all of our Sandy collaborators as well. Just really grateful for that time. And with that, I'll take any questions. Thanks. Yep.
That was a great presentation, and it's the first time in 50 years you made this great homesick.
Well, I'm sorry for the homesick, but maybe that means it's time to go. Thank you, Dennis. I appreciate that.
Thank you. Appreciate it.
Yeah. Okay.
How did your collaborators come up with like 6,500 collections being a representative enough sample size to provide enough resolution and kind of elucidate the relationships?
It's somewhat arbitrary. Good question, but it was calculated just by what we know about the limitations of our ability to collect as a team across 5 field trips. So we knew that would probably cap our ability. So that was just based on the reality of highest expectation of collecting 1,500 samples per field trip. So we just use that as a calculation to generalize that number. Good question though. Yeah, Peter?
Question. One of them kind of technical, but I'll start with that. With the angiosperm 353, why use that when we got this composite?
Would you mind repeating the question?
Peter's asked why we choose to use the angiosperm 353 data set when we already have this other specific probe set for the family composite. Great question. The reason is we're trying to create, I guess, what you would call interchangeable data sets. We're trying to generate robust data sets that are able to be compared from different researchers in a cohesive way. So there are other researchers around the world who are only using Angiosperms 353. There are others who are only using Composite 1061. If we sequence loci from both of those data sets, we'll generate data that will be comparable for any other, we hope, research that has been done using this technique by other researchers around the world. We're also following the lead of our collaborators at Kew. Kew in 2015 launched this program called the Plants and Fungal Trees of Life. And as part of Plants and Fungal Trees of Life, Kew is generating sequence data for one species of every genus of plants on Earth. And the sequence kit they're using is actually the combined Angiosperm 353 and the Composite specific data set. So they're generating sequences for every species of plant using Angiosperms 353. But then if it's available, if there's a more specific bait set available, they're also combining with that data the more specific bait set. So this gives us a complete and more comparable base of data to use for our work. Good question.
Thanks. Another one is maybe not, but it's sort of that. If you're in the field looking for these ironweeds, do you have to ever look at the really technical characters like the styles and things like that, or can from a distance can you just say, oh, that's an ironweed and go from there?
You can usually identify them as an ironweed easily. It's getting them to the correct genus that's a challenge. And one of these, I can give an example. Parapolydora gave us some trouble. One of the species, Parapolydora gerardii, we collected on like day 6 as we were leaving the Barberton Range and going to Natal province, KwaZulu-Natal. And it was just growing on the side of the road. And it was almost an afterthought, like, oh, what the heck is this? I didn't even see it. And we collected it. And we were trying to figure out, okay, what is this thing? Because it wasn't on our list of expected species from this area. And we saw it has these, you can kind of see it here. Actually, it's a great example. We had to actually, when we got back to the herbarium, consult the literature and other specimens to make the proper identification. When we collected it, we noted that the filaries, they're very narrow and almost caudate, so they're almost tailed, right? You can see they're very narrow and tailed. You can see on the side of the backdrop of the newsprint here. In the literature, in the description of this species, it says that these filaries are always recurved, so they start to bend backward. And we didn't see this in this species, and so we wondered, like, okay, that is listed as a key diagnostic character for this species, and we're not seeing these filaries recurve. We're not seeing them bend back. So what is this thing? Is this in a different species? Well, it just so happens that whoever wrote that particular descriptive note had only looked at a limited amount of material, and it's likely that those filaries can exist in a number of different states. So sometimes they'll recurve backward, sometimes they'll stay erect like that. When we got back to the herbarium, we looked at other sheets for the species, and we realized there are other collections that don't have the recurved filaries. And so we realized, sure enough, this is Gerardia.
What's in common?
Oh, yeah.
I have one from the chat.
Uh-huh.
Great presentation. You mentioned worms as the database with modifications. What were some of the other databases you considered, and why did you settle on worms?
Great question. The reason we used worms is really easy to answer. In 2015, I think, the LifeWatch Institute, who manages worms, contacted Vicky Funk. They contacted her because the Global Composite database in its previous life existed as the Global Composite Checklist. And this was managed and maintained by Landcare Research Group in New Zealand. It was maintained as a database, online, freely accessible, for free. They decided no longer to fund that project in New Zealand. And somehow the team at the LifeWatch Institute, which is based in Belgium, heard about that and asked Vicky if she would consider moving the data in the global composite database checklist to the worms architecture, rename it, and hand it over to the management staff of the LifeWatch Institute who runs worms. There were no strings attached. They offered to fly a number of members of TICA to their headquarters in Ostend, Belgium, right on the coast at the Flanders Marine Institute. It's the oldest marine biology institute in the world, and gave us a training for about 4 days in Ostend. So I can't thank the LifeWatch Institute enough for their generosity and for their commitment to supporting these taxonomic resources. That being said, alternative options for hosting the global, for hosting this taxonomic data, I really don't know what else we would consider. I've seen a number of other projects that create their own database, and I do know of other alternatives, but this almost seemed too good to be true. The offer from the LifeWatch Institute. LifeWatch Institute is federally funded from the Belgian government. It's a permanent resource. I mean, I encourage other people to consider contacting the LifeWatch Institute. I'll tell you that colleagues, our co-PI, Isabel Laragon, she's actually a specialist in sedges, Cyperaceae. And she recently reached out to the LifeWatch Institute to ask if they would host the taxonomic data for Cyperaceae. And they said, yes, this is what it would cost. They applied for a grant, they got it. They actually applied for 2 grants, they got both of them. And so they're going to be developing their taxonomic data for Cyperaceae in the same architecture. So this is an awesome group. They have a full team of data management support staff who answer me at like midnight on Saturday and stuff. I don't expect that, but I'm grateful. So if you're wondering where to put your data, please consider the LifeWatch Institute. I just can't say enough good things about them and their staff. They've been amazing. Great question. Peter?
Is there time?
Yeah.
Okay, I was go back to genera, because I was really curious about the Harold Robinson's seemingly quite bold paper, maybe I'm wrong, but in narrowing quite a bit Vernonieae, but at the same time not dealing with the rest of the shrub. So how can one do that? I mean, was that justified to do that? I got a serious question. Was that justified to do that and then leave the rest of them hanging? But then it seems when you go in the field, you can recognize these other things and you're calling them genera and you seem to have them circumscribed. So maybe you could just, if you could kind of, it does seem to be, maybe just.
That's a great question. I thought about putting a slide, and I even started to make it that showed a timeline of taxonomic novelties in Vernonieae that sort of shows the record of the origin of the tribe through present day, and especially shows the description of new genera, many of which have been described by Harold Robinson as a result of his work in 1999. Whether that is whether his approach is, I think I would say it was controversial at the time what he did, shrinking the size of Vernonieae down to just 20 species. That being said, I always enjoyed talking to Harold because he said, you know, I've done a lot in my career and I've made some bold and sometimes foolish decisions, and I usually shrug the foolish decisions off because that just comes with the territory of doing this work. But I also have to smile to myself for all of the decisions I've made that have turned out to be correct. And I have to give him credit. His work has held up from the pre-molecular day to the modern era where we're using DNA sequence data to test hypotheses about classification of different plant groups, especially in composite. And Harold has a lot more in the win column than he does in the loss column, I'll tell you that. What you're asking, whether he was doing it a little bit too soon before we had the data, I would say no. We did have data from a number of works by especially Sterling Keeley, who used to be, she's a retired professor from the University of Hawaii, Manila, who was the first person to really look at using DNA sequence data to reconstruct relationships in Vernonieae. And immediately she knew, it was clear from her data that Vernonieae was not a monophyletic group. We also know that even before that, another researcher, Sam Jones, who is the curator at the Herbarium of the University of Georgia, he utilized pollen to identify groups in Vernonieae, and he also looked at chromosome data to identify groups in Vernonieae. And what he also found was that Vernonieae might not actually be monophyletic. As time goes on, specialists started to work with this group. It wasn't just Harold who started to name and describe one-off genera. There was a whole community of other composite specialists who were doing this at the same time. And so this history of recognizing these distinct genera in this tribe goes back hundreds of years. We have extremely distinct things like Stokesia, which is in Vernonieae, and it's embedded smack dab in the middle of the old definition of Vernonieae. So, I mean, Stokesia is a completely distinct genus. It's monotypic, it's from the southeastern United States, and unlike 99% of all species of Vernonieae, it has radiate corollas, so it has ligulid corollas. So as opposed to just disc florets.
So you're kind of like resurrecting a lot of old names that are already available for these other things that were placed in Vernonieae and like the Old World, for example. They already have like a genus. Are there any that are just described for Vernonieae that are unplaced as the genus? That's a real nasty part.
And I mentioned that a little bit earlier, but it was brief because I had to go quickly. There's still 200 species that remain mostly in Central and Western Africa under the name Vernonieae. And we know they're not Vernonieae. And we need to figure out what in the world they are, because many of these things have only been collected a handful of times. It's going to take a lot of work to sort this out, but we need to figure out what the actual subtribes in this tribe are. Then we need to figure out what the genera actually are. And our preliminary data from Sterling Keeley's last molecular phylogeny shows us that none of the subtribes that are currently recognized in the Eastern Hemisphere Vernonieae are monophyletic. They're all para or polyphyletic. So we have to completely rethink the subtribal classification of Vernonieae, as well as the, sorting out what the individual genera are. But there are some really handy tools we can use. A lot of the reason for many of these unplaced orphan species is simply due to the fact that Harold was kind of the one person doing this work, and he didn't travel much. He was based at the Smithsonian, and most of his work was based on material that was sent on loan, and so he had access to limited material. The US National Herbarium doesn't have a lot of material, especially from Africa, whereas other herbaria do. Missouri, Paris, Belgium, you can imagine, Kew. And so Harold was just kind of working with what he had. And I suspect that a lot of these monotypic genera that he described may stand the test of time and may grow over time as we start to look at more material and see other taxa that are similar to and maybe the same genus as these things that he described from just one single species. But if you look, even here, you'll note like Gymnanthemum is an old recognized name from Cassini. Cassini is basically the father of compositae in many ways, whose tribal classification holds to this day for many of the tribes that we recognize in Vernonieae. So Cassini's work was foundational, and it still holds today a lot of what he described. We also have a number of Linnaean names, Athelia is a Linnaean name. So these are genera that were described long ago. Subsequently, taxonomists said, you know what, there's too many species, they all kind of look the same to me, let's just put them in Vernonieae. No, they were wrong. These were distinctive genera, they weren't all Vernonieae. And so we're just now trying to carefully go through that historical record to piece this back together. Here's another Oocephala, was originally described by Sam Jones at University of Georgia. So Harold resurrected it. So you have numerous instances where you have these old names that have been recognized, and it's just a matter of carefully going back through these archives to understand the taxonomic history of this group.
With that, we are 15 over, so if anyone